So much of the content in this course is going to focus on cellular mechanisms. So I think it's very necessary that we review a little bit the structure of a cell. To start off with, we want to talk about what makes something polar. So what we have here is an oxygen atom and two hydrogen atoms. If we look at an oxygen atom, just like all atoms, it's going to have the same number of protons as it does electrons. So there are, if you look here in the nucleus, there are going to be 8 protons, which have positive charges. And in the shells, there are also going to be 8 electrons, which have negative charges. There's always 2 electrons in the first shell. And then if you look at the oxygen atom, this is, again, true for every atom. If they have 2 electrons, you're going to find 2 electrons in the first shell. And if we look in the second shell of the oxygen atom, there is 1, 2, 3, 4, 5, 6 electrons. Okay, which makes sense, because if we add up the first 2 electrons in the first shell, plus the 6 electrons in the second shell, that gives us 8 electrons. And we know that in the nucleus there are 8 protons. Okay, there's 8 protons in the center. Okay, so the oxygen atom, just like all atoms as they start off with, are neutral. They have just as many positively charged protons as they do negatively charged electrons. Now, what's true for the oxygen atom is it would really like to have 8 electrons in its second shell. That is also going to be true for any atom that has ultimately a second shell. But you can see that the oxygen atom actually only has 6 electrons, and it would like 8. So keep that in mind. Now what we see are 2 hydrogen atoms. Okay? If we look at the hydrogen atoms are the simplest type of atoms. There's 1 proton in the center, right? 1 proton, which has a positive charge, and there is 1 electron in the second shell. So, of course, just like, again, every atom to start off with, hydrogen atoms are neutral because they have 1 positively charged proton and 1 negatively charged electron. But again, that second shell, it is true of all atoms. In their second shell, they would actually like to have 2 electrons. Okay, so each one of these hydrogen atoms that has 1 proton in the nucleus and 1 electron in the first shell, both of these hydrogen atoms would actually like to have 2 negatively charged electrons in their second shell, when in reality, they only have 1. So look at what we've got here. I'm going to erase this real fast. What we have here is a situation where we've got an oxygen atom that has 1, 2, 3, 4, 5, 6 electrons in its second shell when it would like to have 8, and we have 2 hydrogen atoms that each have 1 electron in their first shell when they would like to have 2. So we can resolve this problem of the oxygen atom actually wanting 8 electrons in its second shell, and the 2 hydrogen atoms actually wanting 2 electrons in their first shell by these 3 atoms coming together and forming a water molecule. So what we're going to end up with is something called covalent bonding, or electron sharing, chemistry 101. What's going to happen is the oxygen atom is going to combine, share electrons with the 2 hydrogen atoms and form a molecule of H2O, right? 2 hydrogens and an oxygen atom, H2O, we're going to form a molecule of water. Now, how have we accomplished that, and why have we accomplished that is ultimately the question. How we have accomplished it is if you look at the hydrogen atom combining with the oxygen atom, they are now sharing electrons. So the atoms have come together through electron sharing. And why has this ultimately been accomplished? And the answer is, well, if you look, by sharing electrons through covalent bonds, what we've accomplished here is now this hydrogen atom actually has 2 electrons in its outer shell, which is exactly what it wanted. And this oxygen atom now has 1, 2, 3, 4, 5, 6, 7, 8 electrons in its second shell, which is what it wanted. So the 2 hydrogen atoms now have 2 electrons in their first shell, and the oxygen atom now has 8 electrons in its second shell. So we've created a very stable molecule now through this covalent bonding. The atoms now have the number of electrons that they would like in their respective shells. Wonderful. We have a water molecule that is now made, that has been made through covalent bonds. But the question is, why is the water molecule polar? So let's talk about that now. So ultimately, the reason that this molecule of water is polar is mostly due to the oxygen atom. The oxygen atom is what's called much more electronegative than the hydrogen atoms. What that means is that the oxygen atom tends to pull electrons towards itself, and it creates this asymmetrical molecule of water. And what does that mean exactly? If the electrons have a tendency to get pulled towards the oxygen atom? The answer is you get an uneven distribution of electrons towards the oxygen atom compared to the hydrogen atoms. Okay, so what you have to remember now is that electrons are negatively charged. And if we get an uneven distribution of electrons moving towards the oxygen atom, compared to the hydrogen atoms, what you're going to find is on the oxygen side of the water molecule, there's going to be a slight negative charge. Again, the electrons are unevenly distributed towards the oxygen side of the molecule because the oxygen atom is more electronegative than the hydrogen atoms and pulls the electrons towards it. And this uneven distribution of electrons, which are negatively charged near the oxygen atom, makes the oxygen side of the water molecule negative compared to the hydrogen side, which has a slight positive charge, where there would be, to a degree, a greater distribution of protons. So what makes any molecule polar is when there is an uneven distribution of electrons towards one side of the molecule than another. The way that we typically get there is due to the fact that one of the atoms within the molecule is more electronegative than one of the other atoms within the molecule, and by consequence, the electrons get unevenly distributed near the more electronegative atom compared to the less electronegative atom. And by consequence, on that side where the electrons are more heavily clustered, since electrons are negative, there's a negative charge. And on the side where the electrons are less heavily clustered, there is a slight positive charge. Okay? So any molecule that you ever hear that is polar, right? There's going to be a negative side and a positive side due to an uneven distribution of negatively charged electrons within that molecule. So what happens if we take a whole bunch of polar molecules and we put them together? Okay, what you see here are 3 water molecules, right? We know that each one of these water molecules has a negative side and a positive side. A negative side towards the oxygen atom and a positive side towards the hydrogen atoms. Okay. And if we take a whole bunch of these water molecules and we put them together, we're taking a whole bunch of polar molecules and we're putting them together, what we're going to end up with is what's called hydrogen bonding. Okay, what's going to happen here is this water molecule is going to combine with this water molecule and this water molecule through hydrogen bonding, which really just means that the negative oxygen side of this water molecule, we'll call it water molecule number 1, combines with the positive hydrogen sides of these 2 water molecules. We'll call this water molecule number 2 and this water molecule number 3. You're seeing here that the negative oxygen side, due to that uneven distribution of electrons, of water molecule number 1 is combining with the positive hydrogen sides of water molecules number 2 and number 3. Generally speaking, what does this tell us? It tells us that if you take any molecule that is polar and you put it into water, through hydrogen bonding, that molecule will coexist with water. So an example of this would be like glucose. Okay, I don't have a, sort of a molecular image of glucose here, but what I can tell you about glucose is that glucose is polar. Okay, it's got a positive and it's got a negative side. Glucose is a polar molecule. So what happens to glucose if you take glucose and you put glucose into water? Gatorade could give you, a pretty obvious answer to this, because that's what, well, it used to be more so. Now they use, not as much glucose as they used to. But nonetheless, if you take glucose, which is polar, and you put glucose into water, you're taking glucose, which is polar, and putting it into water, which is polar. Right? So what's going to happen? Glucose is going to be able to coexist, or in other words, bond with the water molecules. Glucose is going to dissolve in the water. So here's what we say about molecules that are polar. If a molecule is polar, it is said to be hydrophilic. What makes a molecule that's polar hydrophilic? Well, first of all, hydrophilic means that it loves water. What that really means is that it can bind and coexist with water. Okay, why? Well, if a molecule is polar, we know that it has a negative and a positive side due to an uneven distribution of electrons within the molecule towards one atom. So if we take a molecule that is polar and put it into water, which is also polar, H2O is polar, then that polar molecule, through hydrogen bonding, will be able to bind with and coexist with water molecules. So that particular polar molecule is hydrophilic. It loves water. It can coexist or bind or dissolve, whatever you want to say, in water. So you'll want to remember why polar molecules are hydrophilic and what that means. So now I want to differentiate between something that's polar versus something that's an ion. Here we have two atoms. We have a sodium atom and we have a chlorine atom. Okay? Without getting into all of the number of electrons and number of protons, if you look at the sodium atom, it only has one electron in its outermost shell. And I can tell you it would like to have 8. Okay? What that tells you about a sodium atom is it does not hold on to that one lousy electron that's stuck out, in this case, in its third shell. Okay? Because it would like to have 8 electrons in that third shell, and it's only got 1. Now, if you look over here at the chlorine atom, it has 1, 2, 3, 4, 5, 6, 7 electrons in its third shell, and it would like to have 8. Right? So this chlorine atom, if it has 7 electrons and it would like to have 8, has a very strong pull to get that last electron in its third shell, which again, where it would like to have 8 electrons. So here's what's going to happen. When the sodium atom and this chlorine atom interact, what's going to happen is the chlorine atom is just going to steal that 1 electron in sodium's third shell. These two atoms are not going to share electrons. What's actually going to happen here is the chlorine atom is just going to snatch and grab that 1 electron in sodium's third shell. Again, sodium can't hold on to that 1 electron in its third shell very well because it would like to have 8. And chlorine, on the other hand, would like to have 8 electrons in its third shell, and it only has 7, so it has a strong pull to finish off the 8th electron within its third shell. So when sodium and chlorine interact, as you can see over here, what's going to happen is sodium is going to lose completely that 1 electron in its third shell, and chlorine, which is now going to be called a chloride ion, is going to gain that 1 electron within its third shell. And what we've actually created here are 2 ions. So what makes something an ion? Ions are not the same thing as polar molecules. Ions are charged, but ions are not polar molecules. So what we have here is a sodium ion and a chloride ion. So what makes sodium, in this case, an ion? The reason that sodium is an ion, first of all, it's what's called a positive ion. The reason that it's an ion or a positive ion or has a positive charge, as we would say, is because it lost that one electron in its third shell. And now if you look, you'll actually find out that sodium has more positively charged protons in its nucleus than it does negatively charged electrons in its shells. After sodium loses that one electron in its third shell, what you'll actually find out is now sodium, the sodium ion, has 11 positively charged protons in its nucleus and 10 negatively charged electrons within its shells. So because it has more protons than it has electrons, protons are positively charged, electrons are negatively charged, since the sodium ion has one more positively charged proton than it has negatively charged electrons, we say that the sodium ion now has a positive charge. This is a sodium ion. Ions that are positive are always going to be ions that lost one or more electrons. And then the reason that they're positive is, of course, because, well, now they have more protons, positively charged protons in their nucleus than they have negatively charged electrons in their shells because they lost some of their negatively charged electrons. And this chloride ion is, well, you could probably imagine it's negatively charged. Again, it's an ion, it has a charge. All ions have a charge, but some ions are positive and some ions are negative. Why would the chloride ion be negative? Well, easy. The chloride ion gained an electron in its third shell. Right? Now it has 8 electrons within its third shell. To start off with, when it was neutral, it only had 7. And now it's gained a negatively charged electron. If you do all the math here, you'll find out that a chloride ion has 18 negatively charged electrons compared to 17 positively charged protons within the nucleus, right? So this chloride ion, after it steals that one electron from sodium and it adds an electron to itself, you'll find out that the chloride ion has now more negatively charged electrons than it has positively charged protons. It has 18 negatively charged electrons and 17 positively charged protons. So since the chloride ion has more negatively charged electrons than it has positively charged protons, we say that chloride ions are negative. Any negatively charged ion, right, and all ions are charged, but if the ion is negatively charged, what that means is that it will have gained a negatively one or more negatively charged electrons, and now it has more negatively charged electrons than positively charged protons. So rather easy to remember. Anytime you see an ion that's positive, you can be certain that it lost a negatively charged electron and now has more positively charged protons than it does negatively charged electrons. And anytime you see an ion that's negative, you can be quite certain that it gained one or more negatively charged electrons, and now it has one or more negatively charged electrons compared to positively charged protons. So what do we say about ions? Ions are also hydrophilic, right? They're water-loving. Why would ions be hydrophilic? Well, if you take a sodium ion or a chloride ion and you put it into water, what do we know? Sodium ions have a positive charge, chloride ions have a negative charge, and water is polar. Okay, water is going to, water molecules, there's going to be negative charge towards the oxygen atom and a positive charge towards the hydrogen atoms. So if you take, for example, a sodium ion, which is positive, and you put it into water, it will be able to bind and coexist with the water. What's going to happen? The positively charged, sodium ion in this example will be able to bind with the negative side of the water molecule. And the negatively charged chloride ion will be able to bind with the positive side of the water molecule. So since sodium ions and chloride ions and anything else that's an ion, potassium, calcium, so forth and so on, sometimes molecules are actually ions. You'll see that with phospholipids. Anytime you've got something that's an ion, it's going to be able to bind and coexist, also called dissolve, in water. So ions are hydrophilic, water-loving, because they can bind and coexist / dissolve in water due to the fact that they're charged. Another way of thinking about this is anything that's charged, whether it's a polar molecule or whether it's an ion, anything that's charged is going to be hydrophilic. Right? It's going to be water-loving. It's going to be able to coexist and bind with water. All right, so not all molecules are polar or ions and thus hydrophilic. We have other types of molecules as well. So what you're looking at here is a carbon atom, and this carbon atom has 4 negatively charged electrons in its second shell when it would like to have 8. And then what I have dispersed around the carbon atom are 4 hydrogen atoms. Okay? If we look at these hydrogen atoms, you know them well. Okay? There's 1 positively charged proton in the nucleus and 1 negatively charged electron in the first shell. All of these hydrogen atoms would like to have 2 electrons in their second shell when they only have 1. And the carbon atom has 4 electrons in its second shell when it would like to have 8. Well, you can probably see what's going to happen here. The 1 carbon atom is going to combine with, through covalent bonding, the 4 hydrogen atoms. And let's take a look at what that looks like. We are going to form what's known as a nonpolar hydrocarbon. Okay, so take a look at this. Through covalent bonding, now the 4 hydrogen atoms are bound to the 1 carbon atom, right, through electron sharing. And what have we accomplished here? Now the hydrogen atoms all have 2 electrons in their first shell, which is what they want, and the carbon atom has a total of 8 electrons in its second shell, which is what it wanted. But what you have to do here is take a look at the molecular structure of this hydrocarbon. It is very nice and symmetrical. There is no uneven distribution of electrons, really, that exists within this hydrocarbon, right? The electrons are nicely distributed throughout the molecule. So there's really no electrons, let's say, that are more distributed on this side versus this side, right? The electrons are nicely distributed throughout the molecule. So overwhelmingly, the molecule is neutral. There's no charge, right? There's no positive and there's no negative side. So what we call hydrocarbons is nonpolar. They don't have a positive and a negative side because the electrons are nicely distributed throughout the molecule. Now, I think this is important to go ahead and say, because this will be relevant to some of our discussions later on. Really, no molecules are entirely nonpolar. Okay? You have molecules that are more nonpolar than others, but every molecule to some degree has a little bit of polarity. This will become important when you start talking about, like Henry's law and some of these other things that can be discussed with gas exchange in the lungs and other concepts. But generally speaking, hydrocarbons are very nonpolar. They don't have a positive side and a negative side because the electrons are very nicely distributed around the molecule. Now what you have to ask is what would happen is if you took a nonpolar hydrocarbon and tried to combine it with water. What would happen if you took any molecule that was nonpolar, didn't have a positive and a negative side, and tried to combine it with water? What you would ultimately find out is that they would repel one another. Okay, so what I'm showing you is trying to combine a hydrocarbon and putting that hydrocarbon next to a water molecule. Okay? You're taking a hydrocarbon, which is nonpolar, and trying to combine it with a water molecule, which is polar. This doesn't work, right? Because there's nowhere for the nonpolar hydrocarbon to bind to and coexist with the water molecule. Okay? There's not a positive and a negative side within this nonpolar hydrocarbon. So by consequence, if there's no positive and negative side, there is no negative side to bind with the positive side of the water molecule, or vice versa. It doesn't exist. Right? So if you take nonpolar molecules and you try to combine them with polar molecules like water, they will repel. So what do we say about nonpolar molecules? We say that nonpolar molecules are hydrophobic. They hate water. Right? They cannot coexist or bind with water. And the reason that nonpolar molecules are really always hydrophobic, can't bind and coexist with water, is because they don't have a charge. They have a very nice distribution of electrons throughout the molecule. There's not a positive and negative side, and thus they cannot bind with either side of the water molecule. So if you take something that is nonpolar and you try to combine it with water, right, or anything else that's polar, they will not be able really to coexist. Nonpolar molecules cannot dissolve in water because they are hydrophobic, right? Because they don't have a positive and a negative side. To give you an example, fatty acids, right, lipids, are made up of hydrocarbons. Fatty acid tails are hydrocarbons. Okay? So if you take fat and you try to combine it with water, they completely repel. They separate into two separate layers, which is ultimately where this whole conversation is going. Take some cooking oil, which is mostly made up of lipids, fat, right? That's a whole bunch of hydrocarbons, and try to combine it into a bottle of water, right? You'll get two completely separate layers. And the reason that you're getting two entirely separate layers is because you're taking a whole bunch of cooking oil, which is made up of nonpolar hydrocarbons, and trying to combine it with water, which is made up of a whole bunch of polar water molecules. You're trying to combine nonpolar molecules with polar molecules, and it's not working. And it can't work. So you're going to want to remember what makes something nonpolar, what makes a molecule nonpolar, and then why are nonpolar molecules hydrophobic? Why cannot they combine and coexist with water? And again, if you want to see an example of that, just pour some cooking oil into a glass of water, and you'll get a great example of what occurs when you try to take nonpolar hydrocarbons, which make up the fat, hydrophobic molecules, and try to combine them with water, which is polar. So the point of all of that introductory chemistry, and for those of you that have taken 6116 with me, you've seen that whole thing before too. What was the purpose of readdressing what makes something polar or ionic and nonpolar and hydrophilic and hydrophobic is so that we could actually discuss the structure of the plasma membrane. It's something that we're going to talk about a lot in this class, which is the plasma membrane and integral proteins. And if you don't understand the concepts of, nonpolar and polar and hydrophilic and hydrophobic, it's just impossible to make any sense of the plasma membrane and then all of the mechanisms that we're going to discuss related to the cell. So for something to be called a cell, it has to have a plasma membrane, a barrier that separates the internal from the external environment. If something doesn't have a plasma membrane, it's not a cell. For something to be a cell, you need typically a nucleus, a plasma membrane, and different organelles on the inside. Okay, but one of the requirements for something to be called a cell is the plasma membrane. So what is the plasma membrane made up of? The plasma membrane is also called the phospholipid bilayer. What it's made up of are phospholipids. It's made up of two layers of phospholipids. So before we discuss the organization of the phospholipid bilayer, there's two layers of phospholipids, I want to talk a little bit about what phospholipids actually are. Again, phospholipids are what are going to make up the plasma membrane of every cell in your body. This is a generic image of a phospholipid. Okay, so the first thing to start off with when we talk about phospholipids, there's going to be this glycerol backbone. Okay, and what you'll see is that the glycerol backbone is typically esterified to 2 fatty acids. Okay, well, the 2 fatty acids that the glycerol backbone are esterified to are made up of hydrocarbons. Fatty acids are mostly what make up lipids. Okay, and fatty acids are made up of hydrocarbons, right? Just repeating over and over and over again. So you can see that the glycerol backbone sort of the phospholipid is attached to these 2 fatty acids, which are made up of hydrocarbons. Thus, the 2 fatty acids of the phospholipid are nonpolar. Oops, my pen's having problems. Okay, the 2 fatty acids are nonpolar. Okay, and the glycerol backbone is also esterified to a phosphate group, as well as the phosphate group is esterified to what's called a head group. Oftentimes, this will be called the phosphate head. It's the phosphate plus the head group, so the phosphate head. Now, here's what we know about the head group. The head group is either an ion or a polar molecule, right? So the head group is always charged. In this case, the most common actual head group is what's called ethanolamine, and ethanolamine is actually an ion. It's a positive ion. It's a molecule that's actually an ion. So this is the major point. If you look at a phospholipid, you're going to find 2 fatty acid tails, which are nonpolar, and then you're going to find a phosphate, again, attached to a head group, and that is going to be the combination, or one or the other. Sometimes the head group is polar, and sometimes the head group is an ion, but regardless, the head group is going to be charged. Now, there are actually a variety of different types of phospholipids. And it's all going, the difference is going to be what is the head group. Okay, so the head group changes. And as the head group changes, the type of phospholipid changes. Not much of the phospholipid structure changes at all besides the head group. Okay, they're really pretty much all the same, except with different heads. And again, all of those heads are either ions or polar molecules. So regardless, if we want to call it the phosphate head, is always going to be charged within all the different types of phospholipids. Now, what you'll also see embedded, within and around the phospholipids are also going to be what are called sphingolipids. You're going to find sphingolipids. You're going to find cholesterol in there as well. And sphingolipids actually are a little bit different than phospholipids. Instead of having this kind of glycerol backbone, you have what's called sphingosine, which serves as the backbone. Sphingosine is still esterified to typically a fatty acid and a phosphate group, which is esterified to a head group. But sphingolipids are not phospholipids. They're different because of the different backbone ultimately that they have. And you'll find out that within the plasma membrane, there aren't just phospholipids, there are also sphingolipids and cholesterol. And then, of course, the question becomes, why in certain cells do we see different quantities of phospholipids versus sphingolipids versus cholesterol, which is also a lipid, by the way? Why do we see that? And it gets really complicated, and this actually, is still to this day an ongoing area of research. But by combining different phospholipids with sphingolipids and cholesterol, you can change the fluidity of the plasma membrane and the permeability and all sorts of other important functional components to the plasma membrane, ultimately, of a cell. Some of that we may get into a little bit as the semester progresses. But mostly I just want to make you aware that if you look within the plasma membrane, mostly what you're going to find are phospholipids, sphingolipids, and cholesterol. And the quantities of each varies, from one cell to the next. All right, so phospholipids mostly make up the plasma membrane. There are sphingolipids and cholesterol that exist in the plasma membrane too, but let's focus on the phospholipids. Why is it called the phospholipid bilayer? Well, because you have two layers of phospholipids, and they are organized like this. There's going to be, let's call this the outer layer. Okay, you're going to find the phosphate heads orientated within the outer layer so that the phosphate heads face what we call the interstitial or extracellular fluid, the ECF. Okay, and then the lipid tails are going to project in towards the cell. The inner layer of phospholipids, we'll call it, is organized so that the phosphate heads face what we know as the intracellular fluid or cytosol, and then the lipid tails project outwards. Right? So this is the phospholipid bilayer. Two layers of mostly phospholipids organized into what I'm going to call an outer layer and an inner layer. Let's repeat this because it's important. Within the outer layer, again, that's not really the correct term, but we can call it that for simplification. The outer layer is organized, the layer that faces the extracellular fluid or is closer to the extracellular fluid, so that the phosphate heads, the phosphate group plus the head group, face the extracellular fluid, and the lipid tails project in towards the cell. And then the inner layer, which is closer to the intracellular fluid, is organized so that the phosphate heads face the intracellular fluid or the cytosol, and the phosphate heads project outwards towards the outside of the cell or the extracellular fluid. Now, why would we want this organization? Okay, well, the phosphate heads, as you know, are made up of both, depending upon the type of phospholipid, a head group that is either polar or ionic. So it's charged. That's all we need to say. It's charged. It's charged. It's polar or ionic. Okay. And the lipid tails are made up of mostly hydrocarbons, right? So they're nonpolar. The whole entire core of the plasma membrane, the core of it, is nonpolar. Look at the entire core of the plasma membrane is made up of lipid tails for the most part, which are made up of hydrocarbons and are thus nonpolar. So look at that. The whole core of the plasma membrane is nonpolar for the most part because it's made up of lipid tails, fatty acids, which are nonpolar, made up of hydrocarbons. And then, why do the phosphate heads within the inner layer face the ICF, the intracellular fluid? And why do the phosphate heads within the outer layer face the extracellular fluid, also called the interstitial fluid? We can differentiate between extracellular fluid and interstitial fluid later on in the class. Why would it be organized this way? Well, because the extracellular fluid is mostly made up of water. What is the intracellular fluid mostly made up of? It's mostly made up of water. The intracellular fluid and extracellular fluid is mostly composed of water. Okay. Well, if the extracellular fluid is mostly composed of water and the intracellular fluid is mostly composed of water, then the intra and extracellular fluid is mostly polar. Okay. The extracellular and intracellular fluid is mostly made up of a polar substance that is water. Okay, so why would we want the phosphate heads within the inner layer, which are also either polar or ionic, to face the intracellular fluid. And why would we want the phosphate heads within the outer layer, which again are either polar or ionic, to face the extracellular fluid? The answer to this is relatively simple. Okay, we want both the internal and external periphery of the plasma membrane to be hydrophilic, right? Because we want the internal as well as the external periphery of the plasma membrane to be able to coexist with the intracellular fluid, which is mostly made up of polar water, and to be able to coexist with the extracellular fluid, which is also made up of polar water. Right? So the reason that we want the internal periphery and the external periphery of the plasma membrane to be made up of polar or ionic phosphate heads is so that these polar or ionic phosphate heads can bind and coexist with the water, polar water, that makes up the intracellular fluid as well as the extracellular fluid. So the internal and external periphery of the plasma membrane is hydrophilic thanks to the polar or ionic phosphate heads. To elaborate a little bit more on this, you would not want the intracellular fluid to be repelled away from the internal periphery of the plasma membrane. That doesn't make any sense. You also would not want the extracellular fluid to be repelled away from, again, the external periphery. It wouldn't make any sense. You want let me erase a little bit here. You want the intracellular fluid and the extracellular fluid to be able to coexist with the peripheries of the plasma membrane. You want the internal intracellular fluid and extracellular fluid to bind with and coexist with the peripheries of the plasma membrane. And that is why the internal and external periphery of the plasma membrane is made up of phospholipids. Of phosphate heads, excuse me. Because they're polar or ionic and can coexist and bind with that intra and extracellular fluid. So then the next question comes into play, which is, why is the core of the plasma membrane made up of nonpolar, right? Nonpolar hydrocarbons, these fatty acid or lipid tails. Right? Why is the core of the plasma membrane made up of nonpolar hydrophobic hydrocarbons? Like, what's the point of that? Right? And the answer, again, is relatively simple. The core of the plasma membrane is going to do a great job of keeping what's inside inside and what's outside outside. How is that going to be the case? How is, due to the fact of the core of the plasma membrane being nonpolar, made up of these hydrocarbons, how is that going to keep what's mostly inside inside and what's mostly outside outside? And the answer to that is most of what's in the intracellular fluid, right, is water. Most of what's within the extracellular fluid is water. And beyond water, most of what's in the intra and extracellular fluid is also molecules that are either polar or ionic. So let's give some examples. Within the intra and extracellular fluid, there are things like sodium, potassium, chloride, calcium, right? This exists in the extracellular fluid too. Sodium, potassium, chloride. There's glucose out here. There are different amino acids, right? Most of what's within the intra and extracellular fluid is either polar or ionic. It's just a fact. Most of what exists within the fluid, whether it's the fluid inside of our cells, the intracellular fluid, or the fluid outside of our cells, the extracellular fluid, is charged. Right? It has a charge. It's either polar or it's ionic. And now you have to imagine what that means if one of these molecules tries to come in or go out. So let's just pick water. That's an easy one, right? What about water? We know water, right, is polar. Water's polar. There are more electrons distributed towards the oxygen side of the water molecule versus the hydrogen side of the water molecule, right? So it has a negative and a positive side. So what's going to happen if water, which is polar, tries to come in from the extracellular fluid into the intracellular fluid? Well, it's not going to be able to, because what's the core of the plasma membrane made up of? The core of the plasma membrane is nonpolar, and it's made up of hydrocarbons, these fatty acid tails, and since the core of the plasma membrane is nonpolar, anything that's polar or ionic is going to get repelled as it tries to come through it. Okay? So if water tries to go in, or if water tries to go out, I don't care which direction it is, it's not going to happen because water is polar. And the core of the plasma membrane is nonpolar. You're trying to combine water, or get water, I should say, past the core of the plasma membrane, which is nonpolar, and it's just simply not going to happen. They can't coexist with one another. They'll repel each other. Let's relate this to anything else. What about sodium? I'll give you an actual example. Sodium is very high outside of our cells, and it's very low inside of our cells. So due to what's called just diffusion, sodium is wanting to come in. It wants to move from the extracellular fluid into the intracellular fluid. And the question is, well, can sodium do that? We know that sodium is a positively charged ion, right? It lost one of its electrons in its third shell, the one electron that it had. So it has more protons than it has electrons, and it's positively charged. So if this positively charged sodium ion tries to get into the cell, it's going to have to get through the core of the plasma membrane, which is nonpolar. Not going to happen. The core of the plasma membrane is nonpolar, and sodium is polar. Sodium is an ion. If you try to move sodium, which has a positive charge, through the core of the plasma membrane, which is nonpolar, then the core of the plasma membrane will repel sodium, and it will prevent sodium from getting from the extracellular fluid into the intracellular fluid. Now let me give you another example. Potassium is a positively charged ion, and it's high inside of our cells. It's low outside of our cells, so it's always trying to get out. Is potassium going to be able to get out? Well, potassium is a positively charged ion. The core of the plasma membrane is nonpolar. They can't coexist, so potassium has no means of doing so. Glucose. I told you glucose is polar. Okay, so what about glucose? What if glucose wants to come in and it's polar, right? It wants to get, if you want to get glucose from the extracellular fluid into the intracellular fluid, and glucose is polar, and the core of the plasma membrane is nonpolar, it's not coming in. Right? The core of the plasma membrane, due to the fact that it's nonpolar, made up of hydrocarbons, will repel the glucose. So this is how the plasma membrane keeps what is inside, and what is outside. It does this because the core of the plasma membrane is nonpolar and rejects most of what's within the intracellular and extracellular fluid from being able to come in or go out. Now, the core concept of the plasma membrane keeping what's inside and what's outside might make sense based on those principles. But obviously, we have all the time things in the extracellular fluid and the intracellular fluid that are polar and ionic that we need to get out of the cell and we need to get into the cell. Right? So the plasma membrane can't be this impenetrable barrier to everything that's within the extracellular fluid and the intracellular fluid just because it's charged. Right? There has to be ways for things to get in and for things to get out. What we have embedded in our plasma membranes or attached to the plasma membrane are what are called integral proteins. Integral proteins, again, are these proteins that exist within or are bound to the plasma membrane of our cells. So let's talk a little bit more about these integral proteins, their organization, and what they can do. So some integral proteins are what are called transmembrane integral proteins. These are integral proteins that span the entire length of the plasma membrane. We're going to talk about those quite a bit. Right? Then we also have integral proteins that only span a portion of the plasma membrane. And then we have integral proteins that don't span the plasma membrane at all. They're just attached through a covalent bond to the periphery of it. And sometimes, as you're going to see in a second, you'll have one of these kind of peripheral integral proteins that are actually attached to a transmembrane integral protein. All right, so we have all kinds of different integral proteins that are termed or defined just based off of how much of the plasma membrane they transverse. And now the goal is to talk about, in basic terms, what these different integral proteins can do sometimes. And then as the semester progresses, we'll go into more and more and more detail. All right, so some transmembrane integral proteins can serve as receptors. So maybe you have a hormone or you have a neurotransmitter that comes over to this transmembrane integral protein and it binds to it. In that case, the transmembrane integral protein is what's referred to as a receptor. And then other times you'll have one of these, which we talked about a second ago, you'll have like a peripheral integral protein that's actually bound to the receptor. So you'll have an integral protein, a peripheral sort of integral protein that's bound to a transmembrane integral protein that's serving as a receptor. The transmembrane integral protein is serving as the receptor. The peripheral integral protein is bound to the receptor, bound to the transmembrane integral protein. Okay? And now this peripheral integral protein that's bound to the transmembrane receptor integral protein can actually serve to activate intracellular pathways. It's involved in all sorts of different cascades and pathways that can influence what happens inside of the cell. That should not make enormous amounts of sense to this point. I'm just, for the moment, trying to explain to you that some transmembrane integral proteins serve as receptors, and then we'll have peripheral integral proteins that serve sort of in activating pathways, intracellular pathways. Some transmembrane integral proteins actually serve as pores, right? They serve as direct passageways for certain ions, typically, to be able to come into or go out of the cell. In this image, you're actually seeing a transmembrane integral protein that's serving as a pore for potassium in order to be able to come in. We'll talk about the details of this transmembrane integral protein, again, also called a pore, in much more detail in the next discussion, which is going to be about membrane transport. But for the moment, please know that transmembrane integral proteins sometimes serve simply as pores for certain ions or different molecules to be able to come in or go out of the cell. And then sometimes, this we're going to talk about relatively soon, we have integral proteins that actually serve as enzymes. This is a transmembrane integral protein that's actually going to serve as an enzyme. And sometimes, as you can see kind of actually in this image, without getting into all the details of this, you'll have a transmembrane integral protein that can serve as both a receptor and an enzyme. Basically, you're going to have some sort of, in this case, it's typically a hormone that comes along and binds to the transmembrane integral protein that we call a receptor. Then it activates the transmembrane integral protein. And the transmembrane integral protein also serves as an enzyme that starts typically phosphorylating things on the inside of the cell. So here you can see sometimes transmembrane integral proteins can serve as receptors and enzymes at the same time. So what's the whole point of this discussion today? The whole point of the discussion is to say every single cell in your body has a plasma membrane. And that plasma membrane is made up of a core of nonpolar hydrocarbons, right? Hydrophobic molecules. And that is what keeps what's outside outside and what's inside inside, because most of what's outside in the extracellular fluid and most of what's inside in the intracellular fluid, it's charged. It's either polar or ionic, and thus it can't get past the core of the plasma membrane, which is nonpolar. Okay? But at the same time, we need things that are polar and ionic in the extracellular fluid to come in, and things that are polar and ionic in the extracellular fluid to go out. So embedded throughout and onto the plasma membrane of all of the different cells in our body, we find different types of integral proteins. These integral proteins serve many different roles. Sometimes they let what's outside come in and what's inside go out. Sometimes they allow what's outside to stimulate the cell in different ways. Basically, there needs to be a means for things in the outside to come in and things in the inside, to go out. And there also needs to be a way for the extracellular fluid, things in the extracellular fluid, to be able to communicate and stimulate the cell. So that's why we have these integral proteins, which sometimes serve as enzymes, sometimes serve as intracellular pathways, sometimes serve as pores, sometimes serve as receptors. All these, these different integral proteins are going to allow certain things out, certain things in, and allow for the external environment around the cell to communicate with the cell. And that's really what we're going to spend this semester mostly talking about, are intracellular pathways. And they're going to oftentimes come back to the discussion of different types of integral proteins. And they're going to be discussed heavily in the next conversation that we have here, which is about membrane transport. We're going to spend some time looking at the different ways that molecules can travel in and out of these integral proteins. And by that, I mean we're first going to focus on the integral proteins that serve as passageways for certain molecules to come in and to go out. And then later on, we're going to start looking more closely at the ones that serve as receptors, enzymes, and intracellular pathways.